back. So this is part 5. I'm Elisa, and I'm going to talk to you about the circulatory system. We're looking at blood, and this is part 5 of hematopoiesis, which we're continuing on with. So part 4 has the beginning of it, and we're going to look more closely at the red blood cell and its formation. Okay, let's get started. Okay, so blood is produced. So we talked a little bit about it requires stem cells, right? So in our case, hematopoietic stem cells, right? So hema meaning like the blood, right? So it's referring to the heme group primarily found in erythrocytes, exclusively found in erythrocytes, and then poiesis, which is the stem cell, poietic meaning like the ability to reproduce all of these new cells over and over again. Multipotent means that this is a type of cell that can rise into many different cell types, right? So you can give a few different outcome stem cells, right? So they're not fully committed at that point. So these multipotent cells, they live in the bone marrow. Now, these stem cells are stem cells, but they are only stem cells for blood. They're not really stem cells for, I don't know, fingernails. They're not stem cells for parts of the liver. They're not, and maybe with scientific intervention that can be changed, but we're looking at what happens naturally within the body. These are committed to become blood cells, but they could become many different types of blood cells. So that's what that means. Eventually, they can give rise to all of the formed elements of blood, right? Not of everything on the body. And they're also called pluri, meaning, plural, more than one type of stem cell. So they give rise to more than one type of adult cells, so erythrocytes or whatnot. We can also call them hemo for that heme group, cyto, cell, so blood cell, blast. So anytime you see blast, think stem cell or like just brand new baby cell from stem cells, essentially. Right? So also hematopoietic stem cells. So these are all the terminologies, and that's just what they mean. And so there's your science words for you. So they multiply in order to maintain their population in the bone marrow and also make the new cells. They also form what we call these like colony forming units, right? These committed groups of cells that are going to form very specialized components of the formed elements or the blood. And so like each one of these like committed or colony forming units are going to really they're going to dedicate themselves to making this particular type of blood cell, right? Or that particular type. Okay. So your erythrocytes, we talked about already. Erythro means red. Cyte means cell. So these are the red cells, right? So mainly their entire function is to do gas transports between oxygen and carbon dioxide, right? That is their jam. That is all they do. They don't divide. They don't live long. They technically, depending on what your definition of a cell being alive is, may not even be alive. It might just be carrier mules. So we'll look at why that is. So we know that they carry oxygen from the lungs to the tissues. We know that they are carrying carbon dioxide from the tissues back out to the lungs, right, as a waste product primarily. And then this is like for metabolism, right? Insufficient, right, anemic, and insufficient red blood cells can cause death within minutes from basically not being able to keep one thing. Remember back in biology? Remember the electron transport chain? Remember how much you hated that? ATP synthase and that whole 9 yards? Yeah, super important because like the one thing that makes this sucker go is oxygen. It's the terminal electron acceptor, meaning it's always pulling the chain and keeping this machine moving, right? It's like the bicycle chain is the electron transport chain of being able to metabolize sugars and produce ATP. And so think of oxygen as the power that does that. So when you don't have enough red blood cells delivering enough of this oxygen around, it's essentially suffocation of tissues, right? Hypoxic state, death within right away. And different rates for different parts of tissues, different tissues. Anyway, so red blood cells are discoid cells, meaning they're kind of disc-like, and we'll look at that. And they have a biconcave shape, like a donut. right? And that's what that's referring to. There's kind of a hole in the middle. They're 7.5 micrometers in diameter, and then they're 2.0-ish micrometers thick at the rim, at the thickest point. And what they do is it's interesting. Like they develop with all of their, and I'll show you a picture because we have those of they develop like a normal cell, right? They have a nucleus, they have little organelles, and then like right before they mature, they actually like eject the nucleus out and some of the organelles and close up, and that's why we get that like kind of concave looking donut shape, is because there's actually a missing element there that sinks in, right? The cytoskeleton maintains that like puffy rim, the donut. So they don't have really mitochondria, and so anaerobic fermentation is how they produce ATP because they don't need a lot. They are born to die. They don't live very long, or they don't remain, I should say, very long after that. So like we said, they eject their nucleus, so they don't have a nucleus, and within that nucleus would be the DNA, which they don't have anymore. And since none of that exists, we're not worrying about doing things like making proteins or definitely not doing mitosis, which is a really good thing because all of that radical oxygen would be causing mutations like crazy. And we don't want that, right? Because that could result in cancer. And so that was kind of a clever way to deal with that issue. So here's what it actually looks like. So here's where you might find like the nucleus and those organelles, but they've been since ejected. The other thing that's really useful is it kind of like keeps all of the parts of the cell that are actually going to come in contact with the tissues and transfer over oxygen or transfer back over, carbon dioxide. It keeps them in close contact, right? So you have that efficiency of transport. So right here, nothing, this part of the cell is not going to touch any type of the tissue. So why would we want it even, a place where any of our heme groups are going to be. So it's actually very strategic that this is a sunken center because it's a non-useful part in the transfer of the gases, right? So now you can kind of see that donut like view. Oh, there they are. There's a photo. Looking good. Looking good. You can see that like collapse in the center. There's a lot of them, right? Nice, a little bendy. Um, so here's this is kind of cool. Wait, let me go back. This is kind of cool actually. So these right here are red blood cells. Of course, they're green. Let me see if I can change my. No. Doot, doot, doot. Okay, so here we go. Red blood cell, right? Okay, this is the side of it. Like it'd be if you took one of these and just, like cut it right in the middle and took a picture. And so you can see how right here was where they're going to come in contact with any of the surfaces. See here, right? And so we're not wasting any of those heme groups in the middle, right? So very, very efficient. And then over here in contrast, here's like a lymphocyte. So it's like a normal puffy cell and it's doing the protein in here. It's got DNA, so it's very busy, um, manufacturing all these goodies. So other things, capillaries. So look at how thin a capillary is. So this speaks to how important it is to have good vasculature. If this is a vessel that is, clogged with, say, there it is, that might be clogged with stuff like, say, animal fats, for example, you might not be able to get a lot of blood supply past here, right? And that's not good. It might get lodged, it's going to break. This is going to be a weaker vessel. So it just, speaks to the importance of having good vessel health. Okay. So erythrocyte form and function. Oh, my green marker back. So blood type is determined by its, oh, you know what? Let's do this. Let me go ahead and talk about this in the next one. So let's go ahead and part 6. We're going to talk about blood types and all this good stuff. In the meantime, I hope you're having a wonderful day, and I will see you soon in part 6. Okay, goodbye now.